So why should we move beyond the neuron in Alzheimer's disease research is a great question. We know now that neurons, so by definition, neurodegeneration in things like Alzheimer's disease is the loss, the progressive loss of neurons and the connections, the synapses between them. But we know they don't work in isolation, so the neurons are supported, the network is supported by glial cells, by the vasculature. And over recent years, we've learned from epidemiology and genetics that these non-neuronal cell types are very important, not just in reacting to the damage in the brain, but we think now that they're somehow contributing to the causes of the disease. So we have several projects in the lab focusing on this. In collaboration with Barry McCall at the UK Dementia Research Institute, we share a PhD student, Maka Sioris, who's looking at how microglia influence synapse degeneration in Alzheimer's disease, and in particular how the ApoE genotype, which is a risk factor for the disease, influences this process. I have another shared PhD student, Monique Hooley, who is with UK Dementia Research Institute Associate Director Giles Hardingham, and together we're looking at how astrocytes influence this process of degeneration in the brain. So yeah, it's a very important time to try and figure out how these genetic changes are causing the brain damage that leads to these diseases. And we know it's not going to be all in the neuron. We've got to look at all the different cell types in the brain. How could our findings impact patients? Well, hopefully, if we understand more about how the brain changes, we'll be able to target that and either prevent or reverse these changes. So I work with several pharmaceutical companies as a collaborator. And it's really gratifying to know that some of the work we've done, while it's been incremental, has really helped inform how therapeutics might be designed. So there are therapies coming up to try and remove A-beta from synapses. There are therapies coming up through pharmaceutical companies to lower levels of the oligomeric species of A-beta and tau, which we helped find out were toxic. So I find that very satisfying that someday the work we're doing on our benches in our lab might really help people with treatments.